Well, welcome everybody to the SIMD FDA webinar. We know we're starting just a few minutes late, but we're allowing some participants to gather. So we just wanted to start with a reminder that this webinar series, this is our third talk, and this is really designed as an opportunity for the Office of Rare Disease at the FDA and the members of the SIMD to have an ongoing dialogue. So if there are other topics that you as a member would like us to cover, please reach out to us. You can reach out to Christy Weiss, who is the Executive Director of SIMD, or the chairs of the Education Committee, so Hong Lee and Mary Morey, or myself, Curtis Coughlin, and we'd love to hear your ideas about the next series. All of these talks are being recorded. They're available on the SIMD YouTube channel. And so if you have not checked out the SIMD YouTube channel, it's a great repository for these talks, their journal clubs, and a bunch of other education information. And as we get started, we are going to have an opportunity to have questions and answers at the end of the webinar. And we really ask you to put those questions and answers down in the Q&A at the bottom. And myself and MK will sort of correlate those questions and ask them to our speakers. So with that, I'm going to hand it over to MK, who's going to introduce our two speakers for today.
All right. So today we'll start off with Linda Jo Bang Jen. She's an associate director for biomedical informatics on the Biomedical Informatics and Regulatory Review Science team in the division of biomedical informatics, research, and biomarker development within the Office of Drug Evaluation Sciences. As a member of the BIRRS team, she supports the work of clinical reviewers and the evidence-based regulatory science decisions of the Office of New Drugs, and serves as the lead for the OND custom medical queries. Linda joined the FDA in 2018 as a medical officer in the Division of Gastroenterology and Inborn Errors Products, and served as a team lead in the new Division of Rare Disease and Medical Genetics before joining ODES. She earned her MD and PhD in cell and molecular biology, completed a combined pediatrics and medical genetics residency, a fellowship in clinical molecular genetics, and directed clinical molecular genetic laboratories for 15 years before joining the FDA. After Linda, we'll have a presentation by Christine Huang. Christine is a senior clinical analyst in the Division of Rare Diseases and Medical Genetics. She joined the FDA in 2012, reviewing drug product applications in the IEM space, first as a clinical pharmacology reviewer and now as a clinical reviewer. Prior to her FDA appointment, Christine performed translational research at the NIH and was an assistant professor at Mercer University. She obtained her PharmD degree at SUNY Buffalo and completed a general practice pharmacy residency and a pharmacogenomics fellowship at St. Jude Children's Research Hospital. Christine is a board-certified oncology pharmacist. So without further ado, I'll hand it over to Linda.
Except I muted. So thank you. And I appreciate the opportunity to speak today. I think we can move to the next slide. This is just the disclaimers that the views and opinions expressed in the presentation are of the presenter and do not represent official policy or position of the FDA, and that there's no financial or non-financial relationships to disclose other than that we work for the FDA. So today I'm going to focus a little bit on just the use of biomarkers as surrogate endpoints as opposed to all different types of biomarkers. I will mention those, but just to be clear that we're going to focus on surrogate endpoints. Next slide. So the drug approval process is outlined in Title 21 of the U.S. Code of Federal Regulations, known as the CFRs. And there are two different approval pathways. One is the traditional approval pathway, and the other is the accelerated approval pathway. Both of these require sufficient information to assess safety and effectiveness of the drug product, and specifically substantial evidence of effectiveness is required in both of those pathways. Next slide. When you look at measuring effectiveness, there's two ways. You can directly measure it, where you're really assessing how a patient feels, functions, or survives. That can be done through various patient-reported outcomes or other clinical outcome assessments. Indirect measurement is more an application for the surrogates, which would predict how the patient feels, functions, or survives, as opposed to actually measuring it. So in the sense for a surrogate endpoint, you would need to have evidence that links that prediction. Next slide, please. The biomarkers can be surrogate endpoints. There are other types of biomarkers, so I just wanted to acknowledge that and go through a little bit about the best biomarker categories, and there's a reference at the bottom of the slide for you. There are different groupings. Some of those biomarkers measure disease presence or status, like susceptibility or risk, diagnostic, monitoring, or prognostic biomarkers. Others measure the aspect of a response to treatment and could be predictive or multi-component or response. Those are more likely to be the ones used as surrogate endpoints, and then there are safety biomarkers as well. So when you talk about the drug approval pathways and you think about traditional approval, if you're using a surrogate endpoint and aiming at traditional approval, you would need to have agreement from the agency that it's a validated surrogate endpoint. And what that means is that the FDA has reviewed and accepts it to predict the clinical outcome that's being specified. And you need to include strong and diverse evidence that that relationship between the surrogate endpoint and the clinical outcome that you're predicting is been established. And again, that would support the traditional approval and would not require additional steps that are required in accelerated approval. So if you go to the next slide, please. So on the other hand, when you're talking about accelerated approval, if you're choosing a surrogate endpoint, that you would have not enough evidence to say that it's a validated endpoint to support traditional approval, but you have enough evidence to make it reasonably likely to predict that outcome of how the patient feels, functions, or survives, then you may have enough evidence to support the surrogate use as a reasonably likely surrogate endpoint for accelerated approval. Often these have very strong mechanistic and/or epidemiologic rationale, and there are a variety of types of evidence that can be applied, which we'll talk about later. But again, this reasonably likely surrogate endpoint would support accelerated approval, and that can include biomarkers. Sorry, next slide, you're good. Evidence is required for the accelerated approval. Like I said, you could support with a biomarker. You could support also with an intermediate clinical endpoint. Those are measurements of therapeutic effect that can be measured earlier than the effect on an irreversible morbidity or mortality. When you look at the biomarkers, you're supporting accelerated approval. So it's this gradient between whether you can support traditional approval or accelerated approval. In some cases, those decisions and discussions have been made a priori before the submission of the application. In some cases, those are made during the review of the application, depending on how much evidence is actually presented and what the results of the review are. And then the other aspect here is just that the biomarker definition from BEST is presented here for you to look at. But again, we went through those classifications of biomarkers for BEST. Next slide, please. So when we're talking about potentially using a surrogate endpoint to support approval, there are multiple options that you can do. There are general type C guidance meetings, and those would involve discussion with the review division about the surrogate endpoint and what's being proposed and what evidence is supported. Those meetings may have further discussion about what additional information may be needed and how that surrogate endpoint would best be used. A type C surrogate endpoint meeting is a specific type of meeting, and that includes a broad group of people involved, the division, but also the biomarker qualification program team and others in the discussion. And for a type C surrogate endpoint meeting, we really want to have clinical information, clinical data to support the use of the surrogate endpoint. And the third is the biomarker qualification program itself. If we go to the next slide, I'll talk more about that. So there are the goals of the biomarker qualification program is really that outreach between the FDA and the stakeholders to identify and develop new biomarkers, to provide a specific framework for the review of the biomarkers for use in regulatory decisions, and then to really focus on what the specific context of use for the biomarker is and have a very clear definition of what's going on. The reasons for that is because once the biomarker is qualified, then it would be able to be used across applications at the agency without further review of the biomarker. So this is a beneficial program to have the biomarkers qualified. Next slide, please. The steps of the biomarker qualification program include a letter of intent, and that's really to request a qualification for a specific biomarker in a specific context of use. As I said, I know that it's frequently the context of use is discussed and revised through discussions. The second is a qualification plan, which describes the biomarker development plans within that context of use that was agreed upon and provide the data on the analytical validation of the biomarker assay. The third step is the full qualification package, and that contains all of the accumulated data to support the biomarker and its qualification for that very specific context of use. And then there's a qualification determination where the FDA will determine whether the biomarker is approved for that use. And as I said, that involves a full review and would then be, the biomarker would be applicable across applications within the context of use. Next step, please. So what does the qualification mean? I've alluded to it a little bit, but it's basically a conclusion that within the stated context of use, the tool or the biomarker can be relied upon to have a specific interpretation and application in the drug development and regulatory review. So once it's qualified, they're publicly available and they can be used in any drug development program, again, within the qualified context of use. And they can be included in INDs, NDAs, or BLA submissions without the FDA having to reevaluate and confirm its suitability. Next slide. So this is kind of the opportunity for us to talk about what kind of information is needed when you're talking about using a surrogate endpoint, a biomarker as a surrogate endpoint. So it's really important that the disease pathophysiology is understood, that the disease progression is understood, how the biomarker fits in with that disease causal pathway, the existence of other causal pathways. So are there other influences that might be changing and altering the disease other than the biomarker that would affect what's going on? Downstream effects. So you may have a biomarker that has additional effects downstream that can change the way the efficacy would be evaluated. The mechanism of action of the drug is important to understand, as well as the distribution of the drug. What compartments are involved, where you're measuring the biomarker or surrogate endpoint, and is it the right compartment that's related to the disease, or is it removed from the primary issue? One example would be if you have a CNS effect and you're measuring a biomarker in the blood, you need to then establish that the levels in the CSF are related and correlated with the levels in the blood. Next slide, please. So evidentiary considerations. There's a lot of things that go into whether the biomarker is suitable or not. Each situation is a case specific and you can't really generalize. So it's very hard to look at one biomarker in one disease and say, well, this worked, so then I should be able to apply this biomarker in that disease, because there are a lot of nuances that go in to the diseases, as well as, again, where the biomarker falls in the pathway, what compartment you're looking at, various different things. That specific context of use that I mentioned before drives the extent of the evidence that will be needed for the regulatory use of a biomarker. And then really it comes down to this convergence of evidence, and all of it should be supportive of the biomarker. In that, there's lots of considerations that go into what should be evaluated, and ultimately it's a matter of scientific judgment. There is a recent publication by Schnook and Sakar that talks a little bit about this, so I provided the reference here on the right, so that you can kind of look at a general context of review of the biomarker. Next slide, please. One of the important things to consider is clinical validation of the surrogate endpoint or biomarker. You need to establish that the biomarker has the ability to predict the relevant clinical concept, and that the biologic plausibility of the relationship between the disease, the biomarker, and the specific outcome that you're trying to predict is supported. It can be supported at different, we're not talking about a validated surrogate endpoint here, this information would go into whether you have a validated surrogate endpoint or a reasonably likely surrogate endpoint. So there's a difference between the clinical validation and the validated surrogate endpoint. So just to be clear, again, this is talking about clinical validation in the general context of the surrogate endpoint. So types of evidence that are important to look at and evaluate and present are epidemiologic, the human genetic and genomic evidence, pathophysiological evidence, translational non-clinical models, or potentially the new alternative methods that can be evaluated instead of non-clinical models. The key point when you're looking at the models is that it needs to recapitulate the human disease. So there are challenges when the model only partially recapitulates the human disease, and there are differences. And if the evidence that you're relying upon is solely from the non-clinical models, that can make the interpretation challenging and be a point where there are issues. Therapeutic effects, dose responses, those types of evidence are very important, human pharmacologic, and then clinical trial data. It's not always required, but it is preferred. And as I mentioned before, for the type C surrogate endpoint meetings, they do require you to have clinical evidence. Next slide, please. The other part that's very important to remember is the analytic validation of the assay to measure the biomarker or surrogate endpoint. Really, you should have performance characteristics established and acceptability levels of reliability, accuracy, sensitivity, specificity, and reference range. What's normal? What are we expecting? What's the change that should be predicted? The other things to really consider and think about, which I've seen some applications not address critically, is what minimal change or particular threshold is required to show that there's a relationship with the prediction of the clinical improvement. So, a small change in a biomarker may not have a clinical significance if it's not been established and shown in some way. The other one is to, as I alluded to before, is to make sure you're measuring in a relevant tissue or compartment or setting up a clear evidence that shows that they're related in a direct way. Next slide, please. The next aspect to consider when you're thinking about using a biomarker or a surrogate endpoint for approval is to really think about whether the accelerated approval pathway is an acceptable pathway. The requirements for using the accelerated approval pathway is that it treats a serious or life-threatening condition with an unmet need. The surrogate endpoint must be measured earlier than irreversible morbidity or mortality or other clinical benefit. This is particularly challenging when the disease course is long and relevant outcomes are infrequent. So those are times where the accelerated approval pathway may be a good option. And then if there are existing treatments, approved treatments, then you need to show a meaningful benefit over those existing treatments. Several considerations go into consideration when you're talking about whether accelerated approval is appropriate or not. The severity of the condition, the rarity of the disease, and the lack of alternative treatments all are weighed in on deciding whether accelerated approval is appropriate. Next. The other things that we need to remember are that the regulatory requirements for substantial evidence of effectiveness are still required for accelerated approval. The other part is a post-marketing confirmatory trial to verify that clinical benefit that's predicted with the surrogate endpoint is required. In many cases, in most cases, it's required to be underway at the time of approval. And what underway is again, something that is up for regulatory discretion based on the severity and rarity of the disease. We also have provisions with accelerated approval for expedited withdrawal of approval if those confirmatory trials are not performed in the ways that have been agreed upon, or if the results are not showing a benefit. You can refer to the, you can refer to the expedited programs for serious conditions, accelerated approvals of drugs and biologics guidance, and that's referenced here on the right. Next slide. We do have regulatory flexibility, and we do apply it particularly in rare diseases. That is true, as I said, with the appropriateness of the use of accelerated approval under Subpart H. The feasibility and the trial design for post-marketing confirmatory trials are really evaluated when we're taking this into consideration. And if a trial is to confirm the clinical benefit isn't feasible, then we often recommend, considering traditional approval methods from the get-go. Differences in the disease, the clinical endpoints, and the study designs are really important in this consideration. And that's, as I mentioned before, it's not easily generalizable when we talk about what's available and what's going to work for a particular disease versus another disease. And the type and quantity of data needed will be different in different contexts and different diseases, and based on risk benefit of the drug and such considerations. Next slide. So overall, I just want to reiterate that a rigorous assessment and appropriate use of biomarkers and surrogate endpoints can hasten drug development for patients with serious diseases and unmet medical need, and a conversation back and forth with the agency to discuss and come to agreement on how biomarkers will be used and what's appropriate, and whether accelerated approval is an appropriate path forward is critical.
So hi, thank you, Linda, for the informative information prior to my talk. So I'm giving you a couple of examples of using biomarker as surrogate endpoint in the IUM space. Specifically, the two examples were, drug products that were approved recently in the for rare diseases. Next slide, please. As Linda mentioned earlier, there is, regulatory standards for approval of drug and biological products for using patients. And the applicant should provide evidence, or the application should provide evidence to demonstrate effectiveness. The substantial evidence requirement for effectiveness typically will ask for conducting 2 adequate and well-controlled investigations. In some cases, but it is limited, 1 large multi-center trial can be used and appropriate for such purpose. I will refer you to the link here that is a guidance showing, what are the requirements for substantial evidence of effectiveness, and you can refer to that. For our talk, we pretty much focus on the other framework that can be used to provide adequate information to demonstrate effectiveness. In certain circumstances, such as in rare diseases with unmet medical need, 1 adequate and well-controlled clinical investigation plus confirmatory evidence, CE, may be sufficient to establish effectiveness. There are a number of considerations that need to check in to determine whether such reliance on 1 trial and CE is appropriate. Some of these factors may include the pervasiveness of the single trial, the robustness of the confirmatory evidence, the seriousness of the disease, particularly where there is an unmet medical need, the size of the patient population, and whether it is ethical or practical to conduct more than 1 controlled trial by 1 controlled trial. There are, a number of data can be used as confirmatory evidence. Just on top of my head, some of the examples include adequate and well-controlled clinical investigation in a related disease area, or using certain type of real-world evidence, such as extensive data providing outcome measures. The last one, that is an example that I will highlight in the next couple of examples, is the use of compelling mechanistic evidence in the setting of well-understood disease pathophysiology or well-documented natural history of the diseases. Next slide, please. Next slide. Oh, sorry. Next slide. So one of the 2 examples that I'm going to talk about here is the first one is Chanodilo for the treatment of cerebral tendinous granulomatosis, CTX. CTX is aware of the autosomal disease recessive disorder caused by a pathogenic variant in the CYP27A1 gene, which encodes for steroids 27 hydroxylase. Deficiency, of the enzyme causes a reduction in the level of bile acid channel deoxycholic acid and subsequent abnormality in bile acid synthesis. The clinical findings include chronic diarrhea, cataracts, tendinopathy, as well as progressive neurological dysfunction. Next slide, please. If you can look at the right side of the diagram, they show you where the metabolic pathway is disrupted in CTX. It may not be very clearly illustrated here, but there is a red box, red text. It showed there was a deficiency in the CYP27A1 enzyme. The deficiency of the enzyme not only, cause, the accumulation of these CDCA, it also inhibit the negative feedback mechanism to inhibit the production of cholesterol. So in that case, there will be accumulation of cholesterol, further leading to accumulation of some of the intermediate, causing accumulation in abnormal bile acid and bile alcohol. It also leads to accumulation of cholesterol, which is cholesterol metabolite toxic to the organ and tissue in CTX. Channeldiol is an orally administered exogenous channel deoxycholic acid. It replaces deficient bile acid in CTX and reduces the production of atypical bile acid and cholesterol. It had been approved in the US for a while for the treatment of radiolucent gallstones in adults and had been used off-label for the treatment of CTX for over 40 years. Although channeldiol had been used as a standard of care, there have been no prospective randomized clinical trials to confirm the treatment benefit of the drug. Next slide, please. In the NDA submission of channeldiol, there was information for one adequate and randomized trial, and that is the RESORT trial. RESORT trial was 24 weeks, randomized double-blind, 2 treatment, and 2-period crossover placebo-controlled withdrawal trial. The trial was conducted in 14 patients greater than or equal to 16 years of age. When compared to placebo, a significant difference of minus 8.5 microgram per ml in plasma cholesterol during placebo treatment, during channeldiol treatment in the placebo periods were observed. Also, additional information about the cholesterol level, which is normalized in 46% of the subject during channeldiol treatment period, compared with only about 7% of the subject during placebo treatment period. Next slide, please. In order to evaluate whether cholesterol is predictive of clinical benefit, the review team has evaluated a number of information from the literature. And after the evaluation, it was determined that cholesterol reduction is clinically meaningful. And the slide lists, some of the following considerations. First, channeldiol accumulated in tissue where CTX caused structural damage or functional impairment. And cholesterol is toxic to tissue where it accumulates, including the CNS. Plasma cholesterol is elevated in CTX patients, and when it is treated with channeldiol, there was a reduction in the level. When the treatment was interrupted or discontinued, there will be a rebound of plasma cholesterol level to a higher level. Finally, in some observational study and case report, plasma cholesterol reduction is associated with improvement or stabilization of CTX symptoms, including seizure, diarrhea, and cognitive impairment. Next slide. There was a confirmatory evidence that support the one adequate and one controlled trial for the approval of channeldiol. So there are information in the literature that show the pathophysiology of the disease is well established, and the mechanism of action of channeldiol is well understood. In addition, natural history evidence confirmed that the changes in the biomarker observed in the RESORT trial deviate from the expected natural history of CTX and results in improvement in symptoms with channeldiol treatment. Next slide. The natural history information from the literature showed that plasma cholesterol, as well as other additional biomarkers, such as urine 23S, phenols, and others, are elevated in majority, if not all, of the untreated celiac patients. And tranodiol treatment decreased plasma cholesterol and all of these other additional biomarkers. Most importantly, this reduction are not expected to occur spontaneously in the natural course of CTX. Next slide, please. After you evaluated all this literature as well as the trial results from the resort trial, the review team concluded that the cholesterol reduction was considered clinically meaningful with strong mechanistic support. Tranodiol was granted traditional approval based on one adequate and well-controlled trial and confirmatory evidence. Next slide, please. The second example that I'm going to go over is the approval of nitisinone for the treatment of alkaptoluria. Alkaptoluria is a rare autosomal recessive disorder that is caused by pathogenic variant in the HGD gene. Genetic mutation in this gene cause a deficiency of homogeneous 1,2-dioxygenase HGD, which causes the metabolism of homogeneous acid HGA. Some of the major features of the patient is that they have debilitating arthritis in the spine as well as larger joints that is caused by a process called onchronosis. Onchronosis is a process that is the deposition of pigment in the connective tissue in the tissue, causing the alteration in the material property of the cartilage, further causing arthritis and rupture of tendons and ligament. There are other manifestations of the disease, including manifestation in the cardiac valve, renal and prostate stone, as well as hypothyroidism. Next slide. This diagram illustrates the metabolic pathway that is involved in AKU, as well as how nitisinone works. Let's focus on the red boxes, which shows the pathophysiology of AKU. AKU is caused by deficiency in the HGD, which is shown inside the red box, and it inhibits the conversion of HGD into its metabolite, causing an accumulation of HGA. HGA is further oxidized to benzalkonium acetic acid, BQA. And BQA is further polymerized to a pigment that is deposited in the tissue, causing the damage to the joints. Next slide, please. Nitisinone is a 4-hydroxyphenylpyruvatic dioxygenase enzyme, which is a competitive inhibitor of this enzyme. HPPD is upstream of HGA, and it causes a competitive inhibition of this enzyme called accumulation, causes a reduction in HGA, further leading to a reduction in BQA, which is the culprit for the pigment that is deposited in the joint tissue. Nitisinone has been approved to treat hereditary tyrosine type 1, and because where it is located in the pathway, it has been evaluated in AKU and has been successfully approved for this indication. Next slide, please. In the nitisinone program, there was also a conduction of one adequate and well-controlled clinical trial. Trial 0076 was a 3-year open-label, randomized, no treatment control trial in 40 adult patients with AKU. As you can see in the table below, at 1 year, there was about 90% reduction in urinary HGAs from baseline for the treatment group. And the reduction of urinary HGA was sustained over another 2 years. On the other hand, for the untreated group, there was about 2-fold increase in urinary HGAs at both 1 year and 3 years. Next slide, please. To show that urinary HGA is predicted a clinical benefit, once again, the review team evaluate evidence from the literature, and the team concluded that urinary HGA reduction is clinically meaningful. Some of the consideration include the followings. BQA, the oxidized form of HGAs, accumulate in tissue where the disease causes structural damage. HGA is toxic to the tissue where it accumulates. Nitisinone treatment leads to consistent and durable reduction of urinary HGA in subject with AKU. And in observational study, as well as case report, the results show the improvement in clinical outcome in subject in AKU receiving nitisinone treatment. Next slide, please. The confirmatory evidence includes in the package is very similar to that of Chenodilo. The first is that the etiology and pathophysiology of the disease is well established, and the mechanism of action of nitisinone is well understood. There were also evidence of efficacy from other published trials, specifically the SONIA 1 and SONIA 2 trial using nitisinone in adult patients with AKU. Finally, there were non-clinical data in the literature that provide mechanistic support for the treatment effect of nitisinone. Next slide, please. This slide summarizes the results from the SONIA trial, SONIA 2 trial. SONIA 2 trial was 4 years open label, parallel group, randomized, no treatment control trial in 130 adult patients with AKU. At 4 years, the increase in the ECOSIS score from baseline was lower, meaning that the patient has less severe symptoms in the nitisinone group compared with the placebo group. There were also trends towards better outcome in other study endpoints. This include the number of fractures, the number of tendons, ligaments, and muscle ruptures, the quality of life assessment, SF-36, and the range of motion of the joints. Next slide, please. Confirmatory evidence is also available for the SONIA 1 trial. SONIA 1 trial is a smaller trial. It is a dose response study, and it was conducted in 40 patients with AKU. It was a 4-year study, randomized, open label, and there are 4 dose levels, evaluating 1, 2, 4, and 8 milligrams of nitisinone. The study results showed that there was dose-dependent reduction in urinary HGA with near completion reduction, about 90%, and doses at 2 milligrams and higher. Next slide. Finally, the confirmatory action also come from non-clinical data in the literature. There were published literature out there that using both in vitro and ex vivo model showing that HGA promote oncogenesis in cell line as well as in cartilage culture model. Also, information is available in knockout mice. We showed that lowering plasma HGA with nitisinone treatment arrests oncogenesis in the tibio-femoral joints of the mice. However, once the pigment is accumulated in the joints, the nitisinone did not further reduce this pigment in the mice. Summary overall in conclusion, next slide please. Reduction in urinary HGA, which come to a level of about 90% reduction, was considered clinically meaningful with strong mechanistic support. And nitisinone was granted traditional approval based on 1 adequate rank-control trial and confirmatory evidence. So this sums up my talk about the 2 examples. I will refer back to the organizer. Thank you.
Well, I want to thank both speakers for giving us this opening and interesting opportunity to talk about biomarkers. And so we're going to open up the question and answer part. So if you haven't noticed, we have a Q&A at the bottom of the Zoom link. So please type in your questions into that area. One of the reasons that we are using the Q&A option is that Our FDA speakers cannot comment about trials that are ongoing where there's an IND. So if you have a very specific question about a trial you're involved with, that is not a question we can ask in this open forum. So to start the question and answer period off, I'm going to ask both of you a question from one of our participants who just wanted if you could give us a little bit more definition or the difference between a validated biomarker that's needed for full approval versus a qualified biomarker that's already gone through the FDA biomarker program.
So I think that one's for me. And I think that the biomarkers and surrogate endpoints leave a little bit of challenge sometimes in how things fit. But a biomarker that's gone through the qualification program could be a validated surrogate endpoint, or it could be a reasonably likely surrogate endpoint. That's all very based on that context of use that I tried to focus on. So a validated surrogate endpoint would be one that has strong enough evidence to support its use, that the FDA has agreed, and it would support and go for traditional approval. There are different ways to become a validated surrogate endpoint. One is having those discussions beforehand, presenting information to the agency, having the agency agree upon it. One pathway would be through the biomarker qualification program. The others are during the course of the review. It may be that there's decision that the biomarker is being used in a way that is supportive of traditional approval and the strong enough evidence presented in the package, and it could be validated during the course of the review of the application. I hope that helps to clarify.
Well, Linda, I'm going to ask a follow-up question then. Do you have recommendations for our memberships of members when they should reach out in this process of considering a biomarker as a surrogate endpoint?
As early as possible. So surrogate endpoints, biomarkers could be discussed, outside of a specific application. There have been times where, a patient advocacy group has come in and asked about information. There are times where it's discussed in a pre-IND. There can be discussed in an IND, can be discussed at all different levels, but as early as possible is very important. There are examples where surrogate endpoints weren't considered initially, but then during the course of development, they have maybe developed further, and that analytical validation of an assay or a new assay has been developed so that it's now a feasible option, whereas it wasn't beforehand. So again, just as early as possible.
I'll add on to what Linda has said. Additional to giving submitting application as early as possible, try to provide as much information as possible so that we understand the disease better and know how best to evaluate the biomarker in the clinical development program.
So it looks like there was a follow-up question to Curtis's follow-up. How can we request such an early meeting with the FDA?
So I think it depends on what stage you're at in that early. Again, if it's a pre-IND, it may be more of that type C guidance meeting to just kind of get that initial discussion started. If it's a patient advocacy group, then there are additional mechanisms, and I am blanking on what the contact point is for that. I don't know if you remember, Christine, but there's an office that the advocacy groups can reach out to.
So I don't know that pathway as well, but I think you can reach out to the RPM, a regulatory project manager under the Rare Disease and Medical Genetic Division of DVRDRDMG, and ask them which is the best route to go through the process. And typically, you can request that kind of meeting initially even though doing a BIND, especially for rare disease, because for rare disease, usually we have very complex program and we have only one trial. And it's really important to start planning early so that, we get the information that we need during the one trial.
I have a question. Do you foresee the potential for integrating multi-omic biomarker panels into regulatory pathways rather than relying on just one surrogate endpoint?
All right, can you just rephrase one more time? Sorry.
Um, do you see a potential for integrating multi-omic biomarker panels into regulatory pathways instead of a single surrogate endpoint? I know a lot of research is being done into proteomics, metabolomics.
Yeah, so I mean, I think that we're open to discussion and new innovative approaches. So I think that those types of meetings and discussions can start and really evolve as they go forward. And the rare disease group at the FDA is another option and opportunity to have those discussions, some of the programs that they have going.
And this is a great overview. Thank you. And so when we use the patients-oriented, like a reported outcome as a biomarker direct or indirect biomarker, how can you distinguish this is real treatment effects from placebo effect and in rare disease field because we don't have in like a large size of participants? How you weight that and as a meaningful biomarker.
Yeah, I think that involves some of our other colleagues here at FDA and in the clinical outcome assessments group, and really evaluating how that works. And in my experience, there's been some tying back to patient reported outcomes as far as using a survey and trying to ground the result and tie it back to how the patient feels, functions, and survives. So those global assessments sometimes help to do that. But
So there is a specific team evaluate.
Yes,
Okay.
which is not me.
But it does bring up a really good question that we are struggling every day in trying to look at our submission. So we pretty much look at, all sort of information that can allow us to do the evaluation and help it understand the disease and how the drug works and whether there's any correlation of the biomarker with clinical outcome, including the COA assessment. So obviously we know that rare disease has a very limited number of patients. The assessment is really challenging. So that's why we really emphasize to do a well-controlled trial in the presence of probably a best to a placebo group, if not maybe a standard of care, arm, so that we can better evaluate what is the COA measurement, what does it mean.
Thank you.
So there are a couple of questions about specific people to contact and how to, and the Office of Rare Disease. And so I think it's a good opportunity to highlight that we're recording these webinars and they're available on the SIMD YouTube channel. And that our very first webinar with the Office of Rare Disease, we sort of laid out some specific contacts, timings that of when you want to start having conversations, pre-IND meetings, what's the regulatory requirement for a pre-IND meeting. The downside is we didn't record that webinar, but we will make all of those slides available from the Office of Rare Disease to members. I hate to put Christy on the spot in the webinar of how we could do that, but please certainly reach out to all of us on this webinar and we can share those slides. What I will say is there's a lot of names and context on those that people may have changed positions over time, but I believe that the title is still the right person to contact. I don't know if Linda or Christine, you have better guidance than what we got from that initial how to contact various members of the Office of Rare Disease.
I don't have anything additional to add.
Yeah, but that is really one of the purposes of these webinars is we want the membership to feel like they know who to contact and when to contact and establish a relationship. So I'll throw this out as well. If anyone wants to contact me, Curtis Coughlin, as well, I'm happy to help that as part of the SIMD membership.
Yeah. And I think that if you have a pre-IND or an IND, then as Christine mentioned, you have a regulatory project manager and RPM. They should be able to help you get to whoever you need to get to within the agency.
So we have another.
If you don't know the RPM, you know Linda and I, you can email us and then we can direct you to the right person.
Wonderful. Again, one of the great things about these series is that we all can put names together to with the titles and faces, and it's helpful to have those personal contacts. So actually, our last webinar was on the clinical outcome assessments and inborn errors of metabolism. And so we have a question that sort of relates to that. So how closely do the review divisions of the Division of Clinical Outcome Assessments work together? Does the DCOA send a consult to the review division during the review of a qualification submission? Do either of you feel that's a question you could answer?
Well,
So
go ahead,
you're
Chris.
referring to the qualification process, right? So it will be Linda's for the question.
So I would say that often it's the review division that's consulting the DCOA as opposed to the reverse. And I think that whenever there's a biomarker qualification program, then they communicate with and work with the relevant division. So there is a individual biomarker qualification program group. And so those happens to be that the biomarker qualification program and the DCOA are all within the Otis office. So we're all in the same office. So there is communication and discussion and communication through the office director as well. So I think that we all work fairly closely together.
Well, I'm not seeing a final question, so I think maybe I want to take this opportunity to thank our speakers for their time. I again want to emphasize that this is really an ongoing conversation between the SIMD membership. So if any members have a topic that they would really like to be addressed, please reach out to the education committee chairs, both who are here, Hong Lee and Mary Mori. You can also reach out to myself or Christy Wees as well, and we would love to make sure that we're being responsive to topics for the membership. And we hope that you will all join us next year as we hope to have the next one of these webinars in the first half of 2026. So thank you all for attending, and thank you again to our lovely speakers.
Thank you for the opportunity.
Thank you.
Thank you.
Thank you.